that mitochondria, the powerhouses of your cells, have their own ribosomes, protein making machinery? They do. And it has some cool medical implications because ribosomes are more closely related to prokaryotic ribosomes, so like bacterial ribosomes. So sometimes antibiotics that target bacteria, if people, especially if people have certain mutations in their mitochondrial ribosomes, then they can be sensitized to antibiotics, and this can actually cause problems. So I mean, like, not cool in the sense of like cool, but biochemically, what's going on is really fascinating. So let's talk about it, starting with what mitochondria are, what ribosomes are, what the two combine to give you a mitoribosome.
So ribosomes are these complexes of protein and RNA, and they put together amino acids, so protein letters, into these long chains to form a protein. And they do so following the instructions provided by the messenger RNA. So the ribosome kind of like travels along this messenger RNA, and then these tRNA, the transfer RNAs, these tRNAs, they're going to bring the corresponding amino acids that correspond to these codons, so these like three-letter nucleotides, so like three RNA letters are going to correspond to in the mRNA are going to correspond to three RNA letters in the tRNA, and that tRNA is going to bring in the amino acid to get added. You keep doing this, you get this polypeptide. This polypeptide is going to be traveling out of a channel of the ribosome, and then it's going to be folding up to make a protein. Ribosomes are made up of these two big parts. Well, one's bigger than the other. There's the large ribosomal subunit and the small ribosomal subunit. And the large subunit is going to have the peptidyltransferase center, so where the amino acids are going to be like joined together. And the small ribosomal subunit has the decoding center. So this is where like the mRNA meets the tRNA. So these large and these small subunits, there are different names for them in the different types of organisms. And these names are in these like something S, and the S is this like Svelberg unit, which relates indirectly to the size based on like a sedimentation rate. So like the heavier you are, the faster you sink, or the further you sink, but it's not like directly linear. So that's why these numbers don't add up, if that's like confusing you. But when it comes to ribosomes, like prokaryotic, so like bacteria and archaea, they have these 70S ribosomes, which has this 50S and this 30S. Eukaryotic, so plants, animal, fungi, they have 80S, which is the 6 and has a 60S and a 40S. And this is for the cytosolic version, however. So the cytosol is like the main area in your cell. It's like the general interior. This is where all of this translation is happening, all of the other good stuff. But there's actually these compartments called mitochondria inside of the cells. And this is a lot of where the energy making is going to happen. So this is a lot of the ATP generation, especially in this inner membrane of the mitochondria, where you have this electron transport chain. So more on this in other posts, but this is a key part of metabolism, especially of the production of ATP from the energy you get from breaking down molecules through catabolism. So the mitochondria are often considered like the powerhouses of the cells. And they have their own genomes, and they have their own ribosomes. And so what's really interesting is that the ribosomal RNAs, so remember the ribosome is made up of RNA and protein. The ribosomal RNAs are actually encoded in the mitochondrial genome, but the proteins are in the proteins for making the mitochondrial ribosomes are actually, the genes are for that are in the like nucleus of the cell, but they are then made and transported into the mitochondria. And the mitochondria genome only has the instructions for making like 13 or so proteins. And these are going to be made by these mitochondrial ribosomes inside of the mitochondria, but there's only these few that are made in here, and they're largely components of the electron transport chain. So many of these components are made based on mitochondrial DNA by the mitochondrial ribosomes. So what is the deal with these mitochondrial ribosomes? Well, to understand, you kind of have to understand where mitochondria come from. So mitochondria basically there's like this endosymbiosis where ancient ancestor of our cells. So swallowed a bacteria or some sort of archaea, some prokaryote. So the prokaryotic cells don't really have like these membrane bound compartments, but eukaryotic cells do. And we have some of these because like the mitochondria, because this ancient cell swallowed one of these bacteria. And so it got its membrane, and it got its like the stuff that was inside it. And the stuff that was inside it had the genetic information for making one of these ancient prokaryotic ribosomes. And so through evolution and stuff, some things have changed, some things haven't changed, but in the end result, you get this mitochondria having a ribosome that is going to be more closely related to the bacteria, to this prokaryotic one than the cytosolic one is. So what's really interesting is that, because ribosomes are so fundamental, because everything needs to make protein, so everything is going to have these ribosomes. And so this is why they can inherit the instructions for making this prokaryotic ribosome a really long time ago. But the mitochondria don't need everything that the bacteria need. And so you can see by the mitochondrial genome, because it's so small now, it only has like those instructions for a very limited number of proteins and RNAs. Because of this, it's shrinking, it's only taking what it needs, but then it can also expand in other ways. So what's really interesting is that the mitochondrial ribosomes, you'll see they're 55S in the mammalian. They're a little bigger in like the yeast, and there's variations. There's a lot more variation in terms of mitochondrial when it comes to species differentiation. But mammalians have this 55S overall, so the complex of the small and the large subunits. This large one is only 39S, the small one's 28S, made up of these three rRNAs. So it's kind of confusing because sometimes you see it listed as 2, sometimes as 3, but it has these two like rRNAs, more like typically for these other ones have as well, 16S and 12S, and the structural tRNA. So the tRNA is actually part of the structure. And what is really interesting is that these RNAs are a lot smaller than the prokaryotic RNA. And so the mitochondrial RNA, like ribosomes, they actually have a ton of proteins. So they have like 82 proteins, whereas the prokaryotic one only has 54. But you can see that the mitochondrial one that is 55S compared to 70S. So it's a lot smaller. And it's a lot smaller because it's decreased the amount of the rRNA it has, like the size of the rRNA is a much smaller component. So in your prokaryotic ribosomes, you have like 2-to-1 RNA to protein, and mitochondrial ribosomes, you have like 1-to-2 ribosome to protein. So you have a lot more protein. And in fact, there's like 36 proteins that are unique to the mitochondrial ribosome. So really, really interesting stuff. But the core part of the ribosome is going to be the same, as we'll see, as the prokaryotic one when it comes to those core RNA parts, because the RNA parts is really what's helping with the actual catalysis, and it's also where like when you have this channel that the growing peptide is going to be coming out of. This is going to, like all of this stuff is very well conserved between the mitochondrial and the bacteria. And we'll get more into what this means, the implications of this in a minute. But another thing that's different about the mitochondrial ribosomes versus the cytosolic ones is that because they're only implicated, they only need to make a limited number of those proteins, those ones for the electron transport chain mostly. The proteins that it has to make are all membrane bound, or like membrane spanning and that sort of thing. In your cytosol, when you want to make proteins that are bound to that are incorporated into membranes, typically those proteins you have to then you start making the protein and then you target it to like the endoplasmic reticulum, which is this membrane bound compartment where it can actually get made in the membrane, so that it does, and then like butt off and all this stuff. But because these proteins that are used to being in membranes, they want to be in membranes, they're like designed destined to be in a membrane, they have different amino acid compositions than ones that are going to be cytosolic. And so you can think of oil and water not mixing. If you have your amino acids are more like hydrophobic, so more like kind of like they want to hang out with like the oily stuff and not the water. And so then the water is going to exclude them and they're going to be all misfolded if you try to get them to fold in this watery environment of the cytosol. So instead you need to send them to this membrane bound place where they can get made in the membrane. And so in order to do this, typically this is directed in the cytosol. This is going to be directed by like a signal recognition particle. And so there's going to be a signaling sequence in the very beginning when the protein starts getting made. Out of that tunnel is going to come this signaling sequence, and that's going to get recognized by these proteins, and you're going to form this signal recognition particle that's going to then transport it to the endoplasmic reticulum where it can get made safely. But because in the mitochondria, it's making these proteins that are going to be in this inner mitochondrial membrane, they're going to be in the membrane, then the mitochondria themselves are going to be permanently membrane attached instead of having to like send them there. So back to the idea, the implications of being more like prokaryotic-like. A lot of times, antibiotics that we use target bacteria by targeting their ribosomes. And we target their ribosomes because, as we saw, they're a lot different than our cytosolic ribosomes. But because they have similarity to our mitochondrial ribosomes, then this can cause problems with toxicity if the antibiotic is actually targeting in some ways the mitochondrial ribosomes as well. This can be a bigger issue if people have a sensitizing mutation. So for example, this aminoglycoside antibiotics. So this includes things like streptomycin, kanamycin, and like paramycin and that sort of thing, more on how they work in my posts on antibiotic selection. But this antibiotic is going to bind to the ribosome in the 30S subunit and interfere with its like quality control. And this is the binding sites. This is a really, really great review article that I recommend. It goes over the structure and the function of the mitochondrial ribosome, as well as some of the evolutionary stuff and some of the implications like this. So Greber and Bond in 2016, annual review biochemistry, and I'll post the link. But some people have this sensitizing mutation, commonly these like A155G or C1494T mutations. So in this structure that is near where the antibiotic is binding and helping, so this there's this like RNA secondary structure. So the RNAs kind of like fold into this, like you have this stranded thing, you have this double stranded here, you have this base pairing in the bacterial one. But this base pairing is broken up in the mammalian one. So here you have this base pairing in the bacteria that's missing that's not going to be present in the mammalian version. But some people have a mitochondrial mutation that is going to convert a mutation in the gene for this mitochondrial RNA that's going to make it so that now there is a base pair again. So when there's a base pair again, which you can happen if you change one of this or change this, then you're going to get this base pairing, and then you're going to get a structure that's more similar to the bacterial one. And because you have this similar structure in this region, then it's going to provide a similar binding site for this aminoglycoside antibiotics that you wouldn't have in the case of if you didn't have that pairing there. So this can put people at greater risk of aminoglycoside-induced deafness. So this is an example of like ototoxicity. And so this is the reason that like people that streptomycin often isn't used as like a first line of treatment, because if people like for like TB and that sort of thing, it's a small percent of the population that actually has this mutation, but enough of a population to make a serious impact on like a public health impact as well as a personal impact, because it can cause this irreversible deafness. And so that is a medical implication. And there's also some implications with regards to doing some antibiotic selection things in the laboratories and eukaryotic cells. Various different things come up. And there's also efforts to try to design antibiotics that target bacterial ribosomes, but are avoid the mitochondrial ones even more.
Another really cool thing about mitochondrial ribosomes is that they use their own tRNAs, so their own transfer RNAs. So some of the genes in the mitochondrial DNA are for making mitochondrial tRNAs. and they use fewer tRNAs than we do with our cytosolic ones. So there's a lot of redundancy with our cytosolic ones, but there's less redundancy with the mitochondrial ones, which means that the mitochondria has to be kind of more flexible in the tRNAs that it uses so it can use the same tRNA for multiple codons. Of course, it's always bringing the right amino acid, but the ability in the decoding center to kind of recognize different tRNAs. This one tRNA for different codons. So that's one other thing that's a little different about the mitochondria, ribosomes, and protein synthesis.
And so that's just some more of random cool stuff about ribosomes. And I had always wondered about mitochondrial ribosomes and whether they were affected by antibiotics. And so, yeah, apparently they can be, especially if you have a sensitizing mutation. So that was my post. And now back to work.